Hello, welcome to MLS QR, a whiteboard quick review for sickle cell anemia and the hemoglobinopathies. Hemoglobinopathy means a disease state involving the hemoglobin molecule, the most common genetic disease in the world. And it involves a genetic mutation of the chromosome that is subsequently transcribed and translated to a defective hemoglobin chain. The hemoglobinopathies are distinct and in direct contrast with the thalassemias, which are a family of diseases that involve a quantitative defect in the synthesis and assembly of the hemoglobin chains. Sickle cell is the hemoglobinopathy prevalent in sub-Saharan Africa, Arab India, Eurasia, and the Americas, and other parts of the world where the mosquitoes thrive to spread Plasmodium species. Worldwide travel, migration, and breeding make it prevalent throughout the world. The sickle cell gene is inherited as autosomal codominant, where both genes are expressed codominantly, and you'll see the normal beta chain in the trait masking the abnormal mutant beta chain until there is a metabolic stress or infection. In the homozygous state, then you see both abnormal genes being expressed. Why is this apparently harmful gene so prevalent in the world? Normally, tough, resilient, and flexible red cells squeeze through capillaries and sinusoids of the reticuloendothelial system, which filters out old, senescent, misshapen, or infected red cells, or those with inclusion bodies. However, theory states that evolutionary pressure begins when the blood-borne parasite Plasmodium infects the young host with hemoglobin S. The oxygen tension drops, causing the hemoglobin S to polymerize, thereby transforming the red cell to lose its characteristic biconcave shape to form tactoid crystals and sickle cell crescents, thereby making it more easily to dispose of by the reticuloendothelial system, giving the infected child a better chance to mount a specific immune response to the parasite and survive until adulthood to pass his or her genes to the next generation. The dilemma is when a patient with sickle cell anemia undergoes metabolic and respiratory distress, leading to hypoxic and acidic conditions, thereby promoting hemoglobin polymerization, tactoid crystals, sludging, high viscosity, which leads to more deoxygenation and acidosis, and then a vaso-occlusive crisis ensues. And then the capillaries, venules, and arterioles get a traffic jam, which leads to infarction of the organs. Acute chest syndrome with pulmonary infarcts and pneumonias may initiate a sickle cell crisis. The bone marrow gets involved. Necrosis, fat emboli, you have osteomyelitis, and this may progress to an aplastic crisis. Chronic sickle cell anemia and chronic hemolysis may warrant exchange transfusions in a outpatient setting in the ambulatory treatment center where you have 6 units of specially screened packed red blood cells negative for C, E, and K antigens and hemoglobin S negative with an emphasis on fresh units to avoid hemochromatosis of the liver and other organs. What is the molecular basis of sickle cell anemia? Normally, DNA is transcribed to messenger RNA, which is translated to a hemoglobin protein chain. A point mutation in chromosome 11 is transcribed to mutant messenger RNA, which is translated to hemoglobin S protein, where the amino acid glutamic acid is substituted by valine. Valine is hydrophobic, without a charge, less soluble, and a serious molecular structural defect, making it susceptible to form the hemoglobin polymers, liquid crystals, and tectoids under hypoxic and acidic conditions. What are the common findings of sickle cell anemia in the microscope? We know about the crescent sickle cell shape, but also note these other morphological features. Note that the Howell Jolly bodies are DNA remnants that are not taken out by the spleen. Before we go to the next panel, note the hexagonal shaped rod shown below. Going back to sickle cell anemia, also note these other red blood cell features. So when you look at a peripheral blood smear in the microscope, you will recognize the constellation of red cell morphological findings that is characteristic of sickle cell anemia. What other lab tests may help support the diagnosis of sickle cell anemia? The hemoglobin solubility test is also known as the sickle cell test, also known as a sickle cell screening test. And here you use sodium diethionide buffer as a reagent. After adding tiny aliquots of controls, positive and negative, in the patients, we will see if the sodium diethionide buffer, if it will cause precipitates to form, hemoglobin precipitates, and this will block the lines from visibility. After 20 minutes, we see hemoglobin precipitates form and turbidity in the positive control as well as two patients, the sickle cell disease and sickle cell trait, and we can't tell the two apart. What test is next that will be helpful in the diagnosis? Before we go further, a few basics. AFSC, that's the order of migration in hemoglobin electrophoresis for these hemoglobins. Accelerate, fast, slow, crawl. Note the pH 8.4, which is basic gel buffer, as opposed to acidic pH 6.0, and that's used to separate S, D, and G hemoglobins in the basic gel buffer, so that way you can distinguish the two or the three. We must run controls with each run so that you can see that everything is working properly and you can see all the hemoglobins. And don't forget the negative control. By using the positive control as a reference point, we can interpret the results. So what is your interpretation shown here on these electrophoresis? We observe that hemoglobin A and S is seen in the sickle cell trait, but look at hemoglobin D trait. It shows the same pattern. But at pH 6.0, we can see that they are different. So a reminder, note that the hemoglobin D and G travels with hemoglobin S and hemoglobin electrophoresis using basic pH 8.4 gel, but they can be separated using acidic pH 6.0 gel. And hemoglobin E is prevalent in Southeast Asia. And just to mention hemoglobin lipour, as it is mentioned in our ASCP study guide, it is a fusion gene during crossover linkage in meiosis that causes a fusion delta beta hemoglobin chain that is usually asymptomatic. Well, let's see if we're ready for some Q&A. And these questions that follow are modeled after the BOC study guide. They're not exact, but they are similar in their concepts. So let's give it a try. your time and attention in this very interesting topic, and I hope to see you in the next video.